Welcome to The Explainer. We're about to take a tour of the most incredible, most efficient factory you can possibly imagine. And the crazy part? It's running inside of you right now. All right, let's kick things off with a bit of a riddle. What do a butterfly's wing, a curry leaf, and, say, the glistening outer layer of your own eye all have in common? They seem completely unrelated, right? What's the connection?
Well, the answer is actually the fundamental unit of life itself. They're all built by these microscopic little factories called cells. Seriously, these tiny things are the building blocks for, well, every living thing we know. So that begs the question, right? How did we even discover these things? They're invisible to the naked eye. It's a pretty cool story, actually, and it took a long time to piece together.
It all starts way back in 1655. A guy named Robert Hooke is looking at a thin slice of cork under a microscope, and he sees these little empty boxes. He calls them cells. But here's the thing, he was looking at dead cells. A little while later, another scientist, Antony van Leeuwenhoek, he's the first person to see living cells swimming around in a drop of pond water. Can you imagine seeing that for the first time? But it still took like another 150 years for folks like Schwann and Virchow to put all the pieces together into what we now know as cell theory. Basically, everything alive is made of cells, and new cells can only come from old cells.
Okay, so we know they exist. Now, let's get our visitor passes and actually take a tour inside one. And to make sense of it all, we're going to stick with this idea of the cell as a super busy high-tech factory.
First stop, the main office, the headquarters. In a cell, that's the nucleus. This place is the absolute control center because it holds the most valuable thing in the entire factory, the DNA. Think of it as the master blueprints for literally everything the cell needs to do or build.
All right, heading out onto the factory floor, and it is bustling. You see those little machines everywhere? Those are the ribosomes. They're the assembly line workers. They get a copy of the blueprints from the main office, and then bam, they start building the factory's main product, which is proteins.
And where's the actual assembly line? Well, that's this huge winding network called the endoplasmic reticulum. We can just call it the ER. This is where all those new proteins get folded up, tweaked, and modified, kind of like quality control and customization, getting them ready for what comes next.
Okay, so once a product is made, you gotta ship it out, right? That's where the Golgi apparatus comes in. This is the shipping and receiving department. It takes the finished proteins, sorts them, packages them up, and slaps a shipping label on them so they get to exactly where they need to go.
But think about it. None of this would work without power. The lights wouldn't even be on. That energy comes from the mitochondria. These are the power plants of the cell. They take in fuel, like sugars, and convert it into this energy molecule called ATP. It's the juice that runs the entire factory. Last but not least, any well-run factory has to be clean. You can't have junk piling up. That's the job of the lysosomes. They're the recycling and waste disposal crew. They go around breaking down old, broken-down parts and waste, keeping the whole operation tidy and efficient.
So there you have it, the basic tour of our cellular factory. But not all factories are the same. A factory that makes cars is going to look pretty different from one that makes cookies. The same goes for cells. So let's look at the two big models, plant cells and animal cells.
And you can see some really big differences right away. For starters, look at the outside. A plant cell has this super rigid cell wall. That's what gives plants their structure. But an animal cell, it's just got a flexible membrane. And here's a huge one, energy. Plant cells have these things called chloroplasts. They're basically little solar panels. They can make their own food from sunlight. Animal cells can't do that. We have to eat our energy.
For centuries, all we could do was watch. We were like people standing on the other side of the street, just looking at this amazing factory through a window. But what if we could do more? What if we could actually walk inside, go straight to the main office, pull out those master blueprints, and start making our own edits? Well, that what if isn't a what if anymore. It's not science fiction, it's reality. We are living in the age of genetic engineering.
So genetic engineering is basically a toolkit that lets us change an organism's DNA. And the star of that toolkit is a technology called CRISPR. You can think of it like a find and replace function for DNA. It lets scientists go into that blueprint, find a very specific sequence, and then cut it or change it with incredible precision. We can literally rewrite the code of life. So what does that actually look like in the real world? This isn't some lab experiment anymore. This stuff is out there. Let's take a look at a couple of real-world examples that are already approved. First, let's talk about salmon.
This is the aqua advantage salmon. What scientists did was they took a growth hormone gene from a different kind of salmon and put it into this one. The result? It grows to full market size in almost half the time. And the whole idea here is to make salmon farming more sustainable, especially in land-based farms, so we don't have to rely so much on fishing wild populations.
And it's not just about growing things faster. This technology is also a total game changer for animal health, which brings us to our next example, the pig. Okay, so there's this really nasty virus called PRSS. It causes a ton of suffering for pigs and huge economic losses for farmers. Now, think of the virus as a key. To get into a pig's cells, it needs a specific lock. What scientists did with CRISPR was they went into the pig's DNA, the blueprints, and just changed the lock. So now the virus's key doesn't work anymore. The pigs are completely resistant to the disease. That means healthier animals and way less need for antibiotics.
So, think about this journey. In just a few hundred years, we've gone from a guy squinting at a piece of cork and seeing empty boxes to literally being able to rewrite the genetic code inside those boxes. That's an incredible leap in understanding, and it's given us an incredible amount of power. Which leaves us with a really, really big question, maybe one of the biggest of our time. We've learned to read and rewrite the code of life. We're holding the pen. So what should we write next?